We're going to first talk about some updates in clinical trials that are ongoing. There's a lot going on in the clinical trial space. We're just going to hit highlights of a few of these. We'll talk about some highlights from the scientific session that was sponsored by the MGFA that was held last fall. And then we'll talk about a few other research items. And a couple of these were submitted by participants at this session that they specifically wanted to hear about. And I also included here on this line that we won't be talking about the MG thymectomy results or the guidelines, but I just want to take the moment to emphasize that those are the two most important things that have happened recently in the field that directly affect the care of patients. So the first study is for an old drug that's been around for a long time. It's called methotrexate. This study was funded by the Muscle Study Group, which is a group of investigators that are interested in studying muscle diseases, which includes myasthenia gravis. And it was actually funded by the federal government, but the study was conducted by the Muscle Study Group, I should say. And so the objective of the study was to look at whether methotrexate was effective at lowering patients' prednisone doses for patients that were already on prednisone. And so patients, as Dr. Kaminsky mentioned, they were randomized to either get the study drug, methotrexate, or a sugar pill, placebo, that they couldn't tell the difference between. And so the primary outcome was looking at essentially their prednisone dose over the last 4 to 12 months of taking the drug. And essentially, this study showed that there was no difference in the prednisone dose after 12 months. And a lot of the other outcomes that were measured in that study also did not meet statistical significance. So it appears that at this time with this particular study that there was no definitive suggestion of efficacy of methotrexate for treating myasthenia gravis patients. There are arguments about the particular study design in this particular case that I'm not going to get into, but at any rate, in the general treatment of myasthenia gravis patients, most MG specialists would not have methotrexate high on their list. The next therapy is called CVZ-533, and this is a novel treatment that was initially looked at in preventing organ transplant rejection. And so in that particular case, after someone has an organ transplant, particularly if it's coming from a donor that's not related to them, the immune response that's generated against the donated organ can be a problem. And so the immune system gets activated in that particular case, similar to the overactivity of the immune system in autoimmune myasthenia. And so this particular therapy works by preventing the activation of a particular set of immune cells called B cells. And on the next slide, I'll show you a little bit more about that graphically so that hopefully it makes some sense. And so the objective of this study is to look at the safety and tolerability and some evidence of efficacy of this particular therapy in moderate to severe refractory MG patients. And this study is ongoing in Europe and Canada and Russia. It's not active in the United States at this time. And so just to orient you a little bit with the what this drug is trying to do. So we have, of the immune system cells, there are a whole host of different cells, and some of the most important ones are listed up here. So we have the B cells, and these B cells are the ones that spit out the antibodies, the acetylcholine receptor antibodies, or MuSK antibodies in the case of myasthenia. And then we have other cells called T cells, regulatory T cells, et cetera, that are there as well. And so this particular drug is active on the B cells themselves and hopefully ignores the rest of the cells, which is advantageous because it leaves the rest of the immune system intact, hopefully, to do its natural job of fighting off infections. And so what this drug does is we have a B cell here that's resting, and encounters antigen or something in the environment that causes it to be stimulated, which in the case of myasthenia, we'll say is the acetylcholine receptor. And that activates the B cell. And then as a result of that, the cells start undergoing a series of changes, including proliferation. They expand the numbers, and they switch to different types of very specialized cells. And so this particular therapy is aimed at preventing that step, okay? And we'll come back to this figure on the next therapy I'll talk about. So the next therapy is rituximab, which probably a lot of you may be familiar with, hearing about it in myasthenia. So right now there's a phase 2 study of rituximab that's sponsored by the National Institute of Health. And it's again looking at treatment refractory patients. Importantly, this study also includes MuSK antibody patients as well as AChR patients. And similar to the other studies we talked about, the primary outcome is looking at how much people can reduce their dose of steroids at the end of 1 year. And so importantly, this study is, from the last I heard, was almost done completing recruitment. It will continue for 1 year after the last patient is recruited. So probably about a year from now or a little bit more, the results of this study may be known. This study is important because, as some of you know, the MuSK MG patients appear to respond extremely well to rituximab. So it'll be very important for that population of patients to have the results of this particular study. What's a little less certain is how it fares in acetylcholine receptor patients. And hopefully this study will provide an answer to that. So if we go back to our slide here as to what this drug is doing, so again, we have our resting B cell that becomes activated when it encounters the acetylcholine receptor and then undergoes all these changes, including spitting out the bad antibodies that attack the muscle. And so what this drug does is it actually kills all the B cells. So it's called a B cell depleting drug is the class. It was originally developed for treating certain types of cancer. The nice thing about it, though, is that it's been around for a really long time. So we have a very good understanding of the potential side effects and the risks associated with it, which is obviously with new therapies that are being developed, we don't have that body of information. So although it depletes all these B cells, it leaves the T cells, those and some of the other immune cells that I showed, it leaves them intact. So the rest of the immune system is still functional. Okay, and the last clinical trial update is eculizumab. And so this is a trial that has just, or not just, but has recently completed enrollment. And this class of drug is a complement inhibitor. And so in MG, we know that once the autoantibodies, the AChR antibodies, bind to the acetylcholine receptor, it starts off a cascade that involves these proteins called complement. And complement destroys the membrane of the muscle cell and removes the acetylcholine receptors from the surface so that they can't do their job in terms of having effective transmission of communication between the nerve and the muscle and causing the weakness. So eculizumab, what it does is it prevents the cascade of events that activates the complement. And the primary outcome on that study is looking at the activities of daily living. And that goes back to what we talked about earlier today, is this is a patient-reported outcome is the primary outcome for this study, which is a big change in the field. This is also the first trial, Dr. Howard informed me, to complete its enrollment on time in myasthenia from a major, one of the major trials. So that's a big accomplishment for that study team. The results of this study will be presented in a few months in Toronto. So everyone in the community is fairly eagerly anticipating the results of this study. And if it works, it has the potential to be in line to be the first immunotherapy approved for myasthenia. So we'll wait to see. Okay, now we're going to move on to the scientific session. So there were a number of very interesting presentations at this meeting. So this was a meeting that was held in Hawaii. So some very difficult places for us to travel to. But the nice thing about this particular meeting was because we were so far west, is we had a lot of representation from Asia, so from China and Japan in particular. And so we heard about some of the epidemiology and clinical characteristics of MG patients in China, which has not been widely reported to this point. And so the really interesting thing is that they have a lot of juvenile MG patients. So almost half of their patients are under the age of 18, which is obviously very different than what we see here in North America. And another interesting point here is that their patients, it seems that they respond to immunosuppressive therapy at lower doses and less intensity than a lot of the patients, Caucasian patients, say, from North America. So this suggests that there are distinctive features in the Chinese MG population that we need to look into a little bit more. A lot of it probably has to do with genetics. But a lot of those genetics probably have an impact on the immune system. And if we can understand that, why these patients are easier or appear easier to control, that may teach us something about how we can treat patients here as well. There were two studies looking at subcutaneous immunoglobulin and for treating myasthenia gravis. And a lot of you are probably aware of intravenous immunoglobulins, which are pooled immune globulin proteins from thousands of donors that are put together into the therapy that are then infused through an IV into patients. It's usually used for patients who are experiencing an exacerbation or possibly crisis. And usually the results are seen within 2 weeks or so. IVIG is often administered over several days either in a healthcare facility or sometimes at home. One of the study teams was from Canada, where they don't have, which is obviously a big place, and don't have access to a lot of the healthcare facilities in close proximity to where people live. So the benefit of this subcutaneous product potentially is that patients can self-administer it. Okay, so if it is shown that it's feasible to do this and it's safe and as effective as using IVIG, is that this may be an additional option for patients in the future. So there are several studies looking at the feasibility of this approach that are either ongoing or in startup phase now, so you'll probably hear about this in the future. Another study was presented on these two drugs I'm not even going to try to say the name of, but these are new monoclonal antibody products or drugs that are very specific for targeting anti-PD-1 and CTLA-4. And so these are called, this is called a checkpoint blockade drug. And it is being used in very advanced stages of cancer and is approved for very advanced melanoma. And so a lot of those patients don't have many good options at all, very high mortality rate. And so these drugs have made a dramatic change in the survival of those types of cancers. One of the things that has been observed by using these drugs is that people are developing autoimmune diseases on them. And so there are now several cases of myasthenia that have been documented with the use of these drugs. So for the clinicians in the group, it was very important to us because now we need to be aware of this as a possibility when patients are receiving these drugs for other reasons. But it also may be telling us something about the mechanism of autoimmune disease and also particularly myasthenia gravis. And so one of the other things that's interesting is that the major genetic study that was done in North America also found this CTLA-4 as being one of the major genes associated with myasthenia. So this is kind of a work in progress. This is very early in terms of our understanding of what's going on, but it's something that could become very important in the future. Okay, so now we have a couple requests from the audience for things to talk about. So the first is about vaccine therapy for myasthenia. And so vaccines have been under development at various stages for myasthenia for a long time now, and for a while had kind of fallen off the map. This study, or series of studies by Dr. Lindstrom, is encouraging, and obviously some people in the audience are aware of this. And so what they are trying to do is develop a vaccine that would be injected to cause myasthenia to go into remission. And so we'll kind of walk through that for a second. So here we have our motor nerve that comes down onto the muscle and ends in the end plate. And then this end plate is where the synapse is, where the communication occurs. And we have this membrane in the muscle cell. And this is the acetylcholine receptor. And so this is embedded in the membrane. And the majority of patients that have the acetylcholine receptor autoantibodies, the antibodies bind to regions of this receptor, which is shown here. And so in the past, a lot of the vaccination strategies had tried to use parts of this part of the acetylcholine receptor to vaccinate people to develop tolerance to self or tolerance to the acetylcholine receptor, so the immune system wasn't active. And so that strategy is similar. You may have heard of recent successes with peanut allergies, where they're giving people very small amounts of, or incremental amounts of peanut over time, and have had some very good results. So it's kind of a similar concept. So what they're doing in this study is they're taking a rat and they inject them with acetylcholine receptor, okay? And then that gives you a myasthenic rat, or in this case it turned into a mouse, but that's usually not what happens. But so this is the experimental animal model of myasthenia, so, or EAMG. So, and then from here, what they do is they take this portion, which is inside the cell, which the immune system usually doesn't see. They're taking this part of the acetylcholine receptor and then injecting that into the mouse or rat. And so in this case, the results have been fairly dramatic to this point. And so they found that if you take a rat that has chronic MG based in this model, is that, or you could prevent it from occurring, is that you can immunize them and prevent them from getting MG. Once they had it, if you gave them the injection, is that the MG would go away. And then number 3 was after they had been immunized, and then you tried to give them experimental MG again by injecting the acetylcholine receptor again, is that they did not get the disease. Okay. So this is very promising, but the scientific community in literature is riddled with lots of examples of things that worked really well in animals that didn't translate to humans. So we have to take this with some caution, but this is very promising and incorporates a lot of what has been learned about vaccination strategies over the past 10 years. So their next steps are they're going to do some further animal studies. They have to change the formulation a little bit, and then they're going to go into cats next. And so the cats, so I guess there's a naturally occurring myasthenia gravis that is not experimentally induced, that is maybe a better model. So they're going to look in cats to see how it works in cats. Okay, the next audience request was there was a recent report of stem cell transplantation in myasthenia. And I've had this, this question come up several times in clinic as well. So in this report, which is coming out of Ottawa, Canada, they looked at a retrospective series of 7 patients that were treated over a decade who received stem cell transplant for their myasthenia. And so this was a fairly young population, which I think is important to note. And just if people aren't familiar with stem cell transplants, basically you take out someone's bone marrow or blood, and you isolate the stem cells. And so the stem cells are cells that are capable of reconstituting a person's blood system, so the blood cells and the immune cells. And so you take those stem cells and you freeze them, and you save them for later, and then you give people very powerful chemotherapy agents to basically destroy all their blood cells. And then after that, you then take those stem cells and re-inject them into the person, and then they reconstitute their blood system and immune system from those stem cells. So obviously, this is a really big deal. This is not something that should be taken lightly. So the results of this study showed that all the patients actually achieved stable remission, and they had no symptoms and ended up on no therapy. Okay. So from that sense, it was definitely a success. And again, you just need to emphasize these are very severely ill patients with very bad disease, and this was done as a last resort. And it's also important to note that, once you take out someone's immune system, is that you're going to experience lots of problems from that. So a lot of the patients, a third of them had reactivation of virus and viral infections that had to be treated that could be fatal. In this case, they weren't. But and then also some patients also will develop a secondary autoimmune disease as their immune system recovers or reconstitutes. So in conclusion, this may be an option for very, very refractory patients at very selected centers and selected patients. But I think over the coming years, with a lot of the therapies that I talked about in the clinical trial update, is that a lot of those may supplant this as an option for patients. But it's an interesting study. And so the last thing I wanted to mention was the latest American Brain Foundation MGFA Clinician Scientist Development Award winner, who's Dr. Michael Hare. And so he has a very interesting project that is called the Immunosuppressive Cost Unit. And so the idea behind this is to develop a scale that really quantitates and measures the side effects from immunosuppressive drugs. And so currently, the way that we do this is by basically counting in clinical trials. We count the number of times that such and such an event happens. But those events are not really weighted in terms of how severe they are, how much they affect patients. So I'll just give you an example of that on the next slide. So this is just an example from a clinical trial that's been posted. There's no reason to read all of these, but you can imagine that, a serious infection, that occurs in someone that's on prednisone might be different than, say someone that has a headache or so. So what Dr. Hare is trying to do is to try to find value for a series of side effects that are common with immunosuppressive medications, so that we can properly or more precisely understand and weigh the effects that patients may have from various medications that they may be on. So he has several phases to this project, and it's actually already started. So I think it's a very important concept, and it really values the patient perspective on the effect of medications. And the other really impressive thing about this is that if it works, you could apply this methodology to almost any immunosuppressive therapy, starting with steroids, because that's the one that's used very commonly and is associated with a lot of side effects. But you could use it for any immunosuppressive medication, and it could also be applied to other autoimmune diseases as well, or any other disease that uses these types of medications as well. So I'm going to stop there, but I am just going to mention one more thing, which is the PCORI survey that was mentioned earlier this afternoon. So as was mentioned earlier, so this is a grant application that is being led by Dr. Sanders and Dr. Narayamaswamy from Beth Israel Hospital, Harvard Medical School. And so PCORI is a Patient-Centered Outcomes Research Institute. And this is a government-sponsored entity that is exclusively interested in measuring and understanding patients' experience with therapies. And so we feel that this is a really good opportunity for us to understand better how the medications that you take affect you in your on a daily basis and in your everyday lives that we often don't get by talking to you in the clinic. So we've designed this survey, and the idea of it is we'll get the results back from this. We're asking some very focused questions about the medications that you're on and how they affect you and what's important to you. What do you want to learn about your medications? What's important about the side effect profiles of them, so that we can design our grant application to respond to the ideas that you have or the experience that you have with your immunosuppressive medications. So it'd be great if you could fill out the survey. If you don't have them, I think they have it at the front desk. It should take about 5 to 7 minutes to complete the survey. Once that's done, there will be some follow-up focus groups and things like that will further refine our ideas for the study that we'll ultimately submit. And it's also important to note that PCORI Institute, they require that there's representation from patients throughout the planning process and implementation of all the studies. So there will be representation from the MGFA and patients as we proceed with designing the study. So thank you very much.